In the last lecture for this week, we kind of discussed all of the different types of cells in the nervous system, the neurons as well as the glial cells, and we barely touched on some of the neuroanatomy. We're going to be spending the vast majority of this lecture going over that, kind of the anatomy of the brain and what different parts handle. But like any map that you're reading, you need to use terms that allow you to orient. And just like when you're looking at a map, you have north, south, east, west, left, right. When you're going through a city, when you're within the brain, there are terms that neuroscientists use to orient themselves within the actual nervous system. And that's what we're going to be covering today in the next slide. So I don't want you to feel overwhelmed with this image. It's an image from your textbook, but it covers pretty much every term that is used to orient within the brain. First, you should know that all terms are oriented to a midline, this line drawn down the center of the body and the nervous system. So we have two symmetrical halves of our body, two of pretty much everything, two arms, two ears, two eyes, and so you can draw an imaginary line down the middle and orient yourself relative to that. So we have left and right, which you should be quite comfortable with, and then relative to the midline we've got medial, which refers to areas of the brain that are closer to the midline, closer to the center. Lateral are those areas of the brain that are closer to the outside of the brain. When we're discussing distance away from the central nervous system, proximal refers to structures closer to the central nervous system or body parts closer to the central nervous system. Distal refers to the opposite, structures further away from the nervous system. So just to give you an example, your toes are distal to your hip, but your knee is proximal to your hip. It's all relative to the central nervous system. And so same thing here, proximal is closer to the central nervous system, distal is further away. Another two opposing terms that we can kind of use interchangeably are anterior and posterior and caudal. These four terms can be used interchangeably. So anterior refers to in front of, posterior refers to towards the back. So in this case, anterior would be here. Actually, here it is. Anterior is here, posterior is here. Caudal literally means towards the tail. So most animals, like rats and mice that we use as animal models, are on four legs. And so their nervous system is in a straight line from head to tail. So caudal means towards the tail, rostral means towards the nose. So our nervous system is unusual because there's a bend in it. We are upright. So rostral in the brain means towards the nose. Caudal in the brain actually means towards the back of the head because that's where the structures are located, closer to the back of the head. Once we get into the spinal cord, because there's a bend in it, we end up having caudal towards the tail and rostral being closer to the brain. Two other terms are superior and inferior. Superior means on top of, inferior means below. Dorsal and ventral are also opposites. So dorsal refers to towards the back, ventral towards the belly. So here it is much easier to see, towards the back, towards the belly. But again, because of the bend that exists in the nervous system, dorsal, when you're in the brain, refers to the top of the head. Ventral refers to closer to, the chin area, for example. So here's also an image of the ventral towards the belly, dorsal towards the back. But when we're in the brain, dorsal is towards the top of the head, ventral is towards kind of the bottom half of the face. When we cut through the brain, we can actually view the brain through different planes, and there are three different planes that are commonly used to view the structures that are inside the brain. So it's not a big deal to kind of view the brain from the outside. You can see a lot of structures from the outside of the brain, but most of the structures are located kind of deep within the brain. And so we can cut the brain in different planes to view the structures in different ways. So the coronal plane is probably the most commonly used plane in behavioral neuroscience, and it refers to refers to the brain being cut in this angle from front to back. So this is where the nose would be, this is the back of the head, this is the dorsal surface, this is the ventral surface of the brain, and so the coronal plane is kind of shown here. You cut from anterior to posterior or rostral to caudal. I'm going to erase these so we can see the other planes a little bit better outside of my drawings. So we're going to talk, can't seem to get this. Okay, so we're going to talk about the sagittal plane. So as I mentioned to you earlier, the brain and the rest of the body is very symmetrical. There are two halves of the brain. So if you cut the brain right down the midline, you would end up with what's called the mid-sagittal plane. So if you cut right down the center of the brain, you would get a mid-sagittal brain view. And then if you continued cutting kind of from, so this is a sagittal cut. You'd be cutting like this, from medial to lateral or lateral to medial. That would give you the sagittal plane view of the brain. The last one is the horizontal plane. This is seen sometimes, and what you're doing is you're slicing the brain from top to bottom, so from dorsal to ventral or superior to inferior, to kind of use the same term. I like to use analogies with food because I love food, and a great analogy for slicing these different planes of the brain are the way in which we slice bread. So if you've got a loaf of sandwich bread, and you're just kind of cutting slices of that loaf of bread, that would be the coronal plane. That's exactly the plane in which we are cutting the brain to view it when it's in the coronal plane. If you want a mid-sagittal plane or a sagittal plane, you would look at a hot dog roll and you would slice it right down the middle on the top so you can kind of put your hot dog in it. That's the sagittal plane. And lastly, the horizontal plane is kind of like if you're getting a Subway sandwich, you would slice the bread down the side to create a bun, a sandwich bun, and that would be, or a hamburger bun, same kind of plane. That's the horizontal plane. So if that gives you a better view of these planes of the brain, great. But either way, these are the three different planes of view that we can use to see the internal structures of the brain. All right. What we're going to be spending the rest of the lecture is talking about structures that belong to this chart. Now, it might look overwhelming, but let me orient you to what you're looking at. So early in development, the brain is kind of in a 3-division stage. So it's broken down into forebrain, meaning front brain, midbrain, not surprisingly, middle of the brain, and hindbrain, which is the back half of the brain. Collectively, sometimes the midbrain and hindbrain are known as brainstem. We can further subdivide these 3 larger divisions into 5 divisions, and these are fancier words. They are something encephalon, so encephalon meaning brain. And so telencephalon, diencephalon, mesencephalon, metencephalon, and myelencephalon. The trick to remembering which one comes first all the way to the end is if you remember T for top, every other term falls in alphabetical order. And they are listed in order of appearance from anterior to posterior or rostral to caudal. Okay? So we'll be discussing these principal structures in greater detail, but the telencephalon is the one that contains the cerebral cortex, the one that's visible from the outside. Deeper within the telencephalon are 2 circuits of brain regions known as the basal ganglia or and limbic system. So these are collections of brain structures that work together to perform similar kinds of functions. In the diencephalon, we've got the thalamus and hypothalamus. The word hypo means below, so you can imagine where the hypothalamus is located relative to the thalamus. In the midbrain or mesencephalon, we've got the tectum meaning roof and tegmentum. In the metencephalon, we've got the cerebellum and pons, which are actually quite visible without having to cut the brain. And then lastly, the most critical portion of the brain stem is the medulla. Again, we'll be discussing all of these in greater structures, starting with structures of the forebrain, using the telencephalon, and then starting with the cerebral cortex. So the forebrain is the anterior-most division of the three-division stage, as is the telencephalon. So the telencephalon is the anterior-most division of the five-division stage. It's got three major structures that you will need to know: the cerebral cortex, the basal ganglia, and the limbic system. So the cerebral cortex, generally speaking, handles a lot of the higher order functions that you might think of when someone asks you what the brain does. The basal ganglia handles motor functions, and for those of you who might know anybody with Parkinson's disease, the basal ganglia is a structure that's affected in Parkinson's disease, and the vast majority of symptoms of Parkinson's disease involve motor dysfunction. The limbic system handles what's known as motivated behaviors, behaviors that animals or people will work hard to obtain or perform, so things like food, sleep, and it also handles things like emotion and memory function. So let's tackle the cerebral cortex in a little bit greater detail. So the cerebral cortex is the outer bark, the outer structure of the brain. When you're looking at the brain, you see this really convoluted structure that seems to be taking up the vast majority of what we're looking at. And these convolutions exist because the human brain has a lot more surface area, but without having a giant head, we need to increase surface area of the brain, and the way in which evolution has created that is by creating bends and folds. And so the convolutions have terms. Sulci refer to the grooves in the brain. So these are the grooves, smaller grooves that exist in the brain. Fissures are the bigger grooves, and I'll show you some in a second. Gyri are the actual bulges. So gyrus is a singular bulge. Sulcus is a singular small groove. We have two identical hemispheres, one on either side, and a major fiber tract, white matter tract, bridges these two hemispheres, and it's known as the corpus callosum. The internal capsule is another bridge, another fiber tract that exists within the brain. This particular bridge connects the cortex through to the thalamus. The anterior commissure, which also exists kind of in the cerebral cortex area or telencephalon, connects the hemispheres at the ventral level. So the corpus callosum is much more dorsal in structure, right, towards the top of the head, and the anterior commissure can be found, is a fiber tract, can be found more ventrally towards the bottom of the head. There are four lobes of the cerebral cortex. We'll talk about them in greater detail: frontal, parietal, temporal, and occipital. They were given these names solely because of the bones of the skull under which they reside. So there's a frontal bone, parietal, temporal, and occipital bone, and these particular lobes happen to sit just below these skull bones. All right, so here are the different lobes. I'll point them out to you. But before I do that, I wanted to kind of go over sulci, fissures, and gyri. So here is a fissure. It's a deeper groove. This is the Sylvian fissure, okay? And here is a sulcus. It's a groove, but it's much smaller. Any of the bulges that you're seeing here, so I'm going to outline one right here in the temporal lobe. This bulge here is called a gyrus. So the gyrus or gyri are bulges, sulci and fissures are grooves. Okay, so the frontal lobe encompasses this structure here. It's separated by the central sulcus that I'm drawing out here. So the central sulcus divides the frontal lobe from the parietal lobe. There's no clear sulcus separating the occipital lobe from the parietal lobe, but it's roughly around here, right? It's the back half of the brain. So the occipital lobe is the back half of the brain and largely handles functions associated with vision. The parietal lobe that I'm drawing over here handles a lot of the somatosensory structures, sensory information. So what I mean by somatosensory is touch, muscles, body position. That's somatosensory, knowledge of the position of things originating from the body. The frontal lobe handles a lot of the many higher order, what we call executive functions, so like reasoning, decision making. But a portion of the frontal lobe also handles something fundamental such as motor cortex. In a way, it makes sense that the motor function is close to the decision making because a lot of our decisions to move have to be based on goals that we have that we're trying to accomplish. So it makes sense that the frontal lobe both contains decision-making and reasoning structures as well as structures that oversee movement. The temporal lobe, shown here in pink, handles language, emotion, hearing, as well as memory. So we'll revisit these structures, but let's talk about the cerebral cortex. I've kind of pointed them out in this image, but here they are in greater detail as to what they do. Within each lobe, there are specialized areas of function, but you have to understand also that these divisions are not exclusive, that there might be bleeding over a function in other lobes as well. So I'll give you an example. The parietal lobe largely handles somatosensory function, but there are aspects of the parietal lobe that actually combine somatosensory information and vision that are processing, for example, hand-eye coordination, where the body is relative to our ability to grab an object that we're looking at, on a table, for example. So the frontal lobe, as I mentioned, handles motor and higher cognitive functions, things that we called executive functions, decision-making, planning, personality, behavioral inhibition are what I like to call filter, so we don't automatically say what's on our mind and are mindful of the social situation we're in to determine what we should or shouldn't say. The parietal lobe, as I mentioned, handles all information originating from the body, so skin, muscles, body position, and the kind. Temporal lobe handles audition, which is a fancy word for hearing, language, learning, and memory, and occipital almost always handles solely vision. Within the frontal lobe, as I mentioned to you earlier, there are functions that are considered to be executive functions, and they are largely handled by the prefrontal cortex. So the prefrontal cortex is the newest, evolutionary newest structure in the brain. Humans end up having a larger portion of the frontal lobe devoted to the prefrontal cortex, and it's the structure that develops the latest in human beings. So this particular structure does not develop until we're close to 25 years old, and it's responsible for things like planning, decision-making, working memory, personality, as well as behavioral inhibition. So in a way, it's kind of a little bit unfortunate that these critical functions aren't completely developed until we are, well into adulthood. The cortex is organized in layers. So if we sliced the cortex up, this would be the outer surface, the dorsal surface of the cortex, and this would be the area that would be closer to the inside of the brain. There are 6 total layers of the cortex, and these layers have different kinds of cells residing within them. You don't need to necessarily know what they're called, but it's important to know that the cortex is divided into layers, and these layers house different kinds of cells that perform different sorts of functions. So for example, pyramidal cells handle motor function, and they can be found in layer 5. You might find sensory information being handled by stellate cells, which can be found in layer 4. So different layers of cells in the cortex handle different functions. On to the basal ganglia. So the basal ganglia is one of those 2 structures that kind of reside deep within the telencephalon. The basal ganglia handles nothing really but movement, for your purposes. It includes 3 structures. So the basal ganglia is a collection of 3 structures. They are the caudate nucleus, putamen, and globus pallidus. Sometimes the caudate and the putamen are collectively known as the striatum. This is important because an area of the brain that can be found in the midbrain, the substantia nigra, projects or speaks to the striatum, which is in the basal ganglia. What's unfortunate is that in Parkinson's disease, the substantia nigra ends up dying slowly, and so one of the things that the substantia nigra is doing is activating the striatum for motor function. But when the substantia nigra dies in Parkinson's disease, you end up with a situation where it's very, very difficult to perform motor functions. To give you an idea of where in the brain the basal ganglia is, so it's kind of superimposed, but these structures are deep within the brain. So the basal ganglia is found in purple here. They include the globus pallidus and then the caudate as well as the putamen right here. So the putamen is the outer ring shown here, the globus pallidus is the internal ring, and the caudate is the tail shown here. The other kind of critical structure or collection of structures within the telencephalon is the limbic system. It is made up of structures that circle another area that's part of the diencephalon called the thalamus. It's involved in motivated behaviors such as eating, feeding, sexual behavior, affection, maternal behavior. All of those are handled by the limbic system, as well as emotion and learning and memory. So it includes structures such as the hippocampus, which is a structure critical for learning and memory. We've actually touched on this structure in previous lectures. The amygdala, which handles a lot of the negative emotions that we might feel, like aggression, fear. The cingulate cortex, which coordinates perception, emotional perception of events, as well as the septum. And so here we have structures of the limbic system. Just to orient you, the thalamus is shown here in brown, and all of the structures are colored. So we've got the cingulate cortex shown here, we've got the hippocampus in purple, and the amygdala sits like a cherry on top of the hippocampus in red. So we talked about the telencephalon. We're going to tackle the diencephalon, which is the next structure posterior or caudal to the telencephalon. The diencephalon contains 2 major structures, the thalamus and hypothalamus. The thalamus is considered the relay center of the brain, whereas the hypothalamus handles all endocrine and autonomic control of the brain. So again, just to give you an idea, this is a mid-sagittal plane of the brain. So the brain has been sliced right down the middle, and you get a nice view of the thalamus and hypothalamus. The thalamus is shown here in brown, and peeking through is the end of the hypothalamus. And as I mentioned earlier, the thalamus is the relay center of the brain. What I mean is that nothing gets to the conscious perception of the cortex from our sensory systems unless it goes through the thalamus first. So we don't actually pay attention to every sensory stimulus that our brain receives because it goes through the thalamus first before it reaches the conscious perceiving portions of our brain, the cerebral cortex. You can think of the thalamus as kind of like the administrative assistant to the CEO of the cerebral cortex, and kind of nothing gets through the CEO or to the CEO without going through that administrative assistant of the thalamus first. So the next group of structures after the diencephalon belongs to a new division at the 3-division stage. That's the midbrain, or the mesencephalon. The mesencephalon, if you remember, contains the tectum and the tegmentum. The tectum literally means roof. It's the top half of the midbrain or mesencephalon, and it contains 2 important structures that you need to know. It has others that we're not going to worry about, but you need to know these two structures. It's the superior and inferior colliculi. Colliculus literally means little hills, and I'll show you why they're called little hills in a second. But the superior colliculus handles visual reflexes, and the inferior colliculus handles auditory reflexes. So to give you an idea of what I mean by visual reflexes, if you happen to hear, if you're kind of trying to pay attention to this lecture and somebody goes, passes by your screen, in your kind of field of view, even though you're not paying attention to it, you will shift your vision to that object that recently moved. That's your visual reflex. Our brain's constantly paying attention to objects in our visual world, just in case they might be dangerous. So the superior colliculus's function is to pay attention to any changes in our visual world and respond to it by paying attention to that newly changed visual stimulus. The inferior colliculus is the same thing. Anytime we hear a loud sound, our attention diverts to the direction that the sound came in. Again, for the sole purpose of avoiding getting hurt ourselves. So the reason why the colliculi are called as such is so this is the back half of the brain. The cerebral cortex is actually removed, and here is the thalamus, but this is the superior colliculus, and this is the inferior colliculus. They literally look like little hills on the back half of the brain. The other area of the midbrain is the tegmentum. It is the bottom half of the mesencephalon, and there are three colorful structures that you will need to know in addition to one extra one. The periaqueductal gray handles analgesia or pain relief. So if you've ever taken any morphine or any opiate of any sort, or know somebody who has, the way in which opiates work to relieve pain is by acting at the periaqueductal gray. We've briefly talked about the substantia nigra. It's the area of the brain that dies in Parkinson's disease, and it helps facilitate motor function. The red nucleus is also another structure, again, red nucleus, that handles motor function that we'll not necessarily be covering in this lecture, but if motor function is, in this course, but if motor function is something that you're interested in, come speak to me. I'm happy to talk to you about it. So you might be wondering why the substantia nigra is considered a colorful structure, because the substantia nigra literally translates into black substance, so nigra meaning black substance, and therefore it belongs in one of these three colorful structures. The last one that's not colorful but is important and can be found in the tegmentum is the ventral tegmental area, or VTA. It is a critical part of the reward system, and it is the area of the brain that often gets hijacked when it comes to addiction. So drugs of abuse often work to activate the ventral tegmental area so that we can derive pleasure. Typically, we derive pleasure from things like watching a good movie or eating a good meal. This area will be activated under those conditions too, but the kind of pleasure we can derive from drugs of abuse is much greater than what can be achieved through natural rewards, which is part of the reason why it's so easy for some of us to become addicted to drugs of abuse. Again, just to give you an idea of where we are in the brain, so we're looking at the brain through the mid-sagittal plane, and I'm outlining the midbrain. So here's the thalamus, here's the hypothalamus, and just posterior or caudal to the thalamus is the midbrain. The superior colliculus can be seen here, a little bit what's left of it. The periaqueductal gray surrounds what's called the aqueduct that we'll cover later on. The red nucleus, shown here, the substantia nigra, shown here. And again, these are slightly color-coded, but the substantia nigra actually does appear black without any staining whatsoever, which is why it was given the name substantia nigra. And the red nucleus has a slight pink hue to it as well, which is why it's called the red nucleus. Again, you'll notice that neuroscientists and biological psychologists are not very creative when it comes to naming structures or naming proteins. They're very they are very obvious in their names. Okay, so we're moving further away from the top half of the brain. So we're going to be talking about the metencephalon, which includes the pons and the cerebellum. Both these structures can be viewed without having to cut the brain open. So this was the pons, this is the cerebellum. They both belong to the hindbrain within the metencephalon. So the pons is the bulge on the ventral surface of the brain, and it's largely made up of a network of fibers going from the spinal cord to the brain and vice versa. So it's kind of relaying signals from the body or spinal cord to the higher order structures of the brain. So it acts like a switchboard. It contains collections of chemical neurotransmitters, ones that we will be talking about later on in the semester, like acetylcholine, serotonin, which you may have heard of, as well as norepinephrine. It seems to have a role in dreaming. There's a structure of the pons that becomes activated when we enter rapid eye movement stage of sleep, or REM sleep. And so it's thought that this particular structure, or parts of the structure, seem to be important in dreaming. The cerebellum, or little brain, right? It looks very similar to the cerebral cortex because it's got all those convolutions within it. It is a very important structure for sensory motor functions. What I mean by sensory motor is that whenever we walk into the world, you're not paying attention to it, but we're constantly adjusting our movements as needed based on the information coming into our nervous system. And so, if you're playing outside and you accidentally have a ball coming towards you, even if you're in mid-walk heading towards to see a friend, you would move out of the way. You would adjust your behavior because of the sensory information coming in. So it's a sensory motor function that's handled by the cerebral cortex. It also handles coordination. It's responsible for receiving inputs from the spinal cord and the pons, and then calculating the sequences or the order of muscle contractions required to achieve any particular goal that we might have. So the spinal cord sends the cerebellum the body's position, and the pons is sending the cerebellum information from the cerebral cortex. So one of the things that happens in the brain is that opposite sides of the brain handle or control body parts. For example, the left half of the brain controls the right half of the body with respect to movement. The way in which this happens is that neurons may reside in the cortex, but then their axons cross to end up on the opposite side of the spinal cord, and the point at which they cross happens in the medulla in an area called the pyramidal decussation. So most of the crossings that happen in the brain, to control opposite sides of the body, happen in the medulla at the pyramidal decussations. So here's the medulla, of the very last structure of the brain, shown here in blue. It sits in the caudal end of the brain and the brainstem. It is the metencephalon in the 5-division stage and happens to be the last structure of the hindbrain. So the medulla is the most posterior caudal division of the brain. It carries a lot of fiber tracts between the brain and body, but handles a lot of those vital functions that kind of keep us alive and we're not really paying attention to, like respiration and cardiovascular function, sleep, as well as attention. And so here is kind of collectively an image from your textbook of the pons, medulla, and cerebellum together. They make up the met and myelencephalon. We've surprisingly completed all of these structures in this particular lecture. I know it may seem like a lot, and we kind of went through it quickly, but as needed when we talk about different functions, we'll be revisiting many of these structures again, but it's good that you get introduced to it now. What I would recommend you do is create another column here for yourselves where you can write down the functions of these different structures. So having it all together in one sheet of paper might kind of help you digest this information, but we've talked about it separately over many, many different slides, but it might be useful for you to create a chart for yourself so that you have it all together in one piece. That's it for this lecture. Don't forget to submit your reflection for it.